Hi everyone, and welcome to our IF and IHC webinar. My name is Rebecca Northeast, and I am a product manager here at Proteintech. I'll just wait one more minute so that everyone can join on, and then we'll get started. So today is the second day of our immunoassay applications week. Yesterday, I gave a webinar on antibody validation, and if you missed that, we'll be able to send you the recording if you're interested, and it will also be available online. So just looking at who's signing up now, I think we'll be able to get started very shortly. I hope everyone's having a really great week. So yeah, let's get started. So today we're going to be going through the techniques of immunofluorescence and immunohistochemistry. But first, I'm going to start with a very quick company introduction. So if you're not aware of who we are, Proteintech is a leading life science reagent company, and we have products across 3 core product lines. So through our Proteintech brand, we sell our antibodies and immunoassays. And so our antibodies we actually have against 13,000 human proteins, as well as over 400 ELISA kits and our IHC Easy kits. Through our Humankind brand, we sell our cytokines and growth factors, and these are recombinantly expressed in a HEP293 cell line, which is actually a human cell line. So that means that they have native post-translational modifications and glycosylation, which results in higher bioactivity. And then on our Chromatech side, we sell our nanobody-based reagents. So nanobodies, as the name suggests, are much smaller antibodies. And what they are really excellent for is immunoprecipitation of tagged proteins, as well as things like super resolution microscopy. As a company, we like to measure our scientific success by measuring our customers' success. And so we're incredibly thrilled that our products have been cited over 200,000 times worldwide. And we are now one of the top 10 cited antibody companies, and we are in fact the fastest growing company in terms of product citations, which we're incredibly proud of, as it shows our customers are trusting us by purchasing our reagents and then also publishing with them. As a company, we say we are a company that was founded by scientists for scientists, and all our antibodies are made in-house, and we actually have the largest in-house portfolio of antibodies for the human proteome. If you're wondering why we talk about that so much and why it's so important, you can go back and listen to the talk yesterday as we talk about the importance of antibody manufacture and antibody validation in-house as well. And then, so what am I going to talk to you about today? So today we're going to go through, really briefly go through the protocol for immunofluorescence in IHC. Then we're going to, more importantly, do a step-by-step protocol consideration, so figuring out where we can tweak each part of the protocol to best understand how to optimize our workflows and get the best results possible. Then we're going to talk about the reagents that are required for immunofluorescence and immunohistochemistry, and especially why antibodies are so important for this, and the different types of antibodies available to you that you can therefore get the best results. Now, next we're going to go into some detailed troubleshooting, and then we're going to do a Q&A. My colleague Frida is on the line. She is a product manager who has a lot of experience in IF and IHC like myself, and will be able to give you our best tips. And so moving on, first we're going to start with the immunofluorescence protocol. So what is the premise behind immunofluorescence multiplexing? Essentially what this is where you have antibodies that are labeled with different non-overlapping fluorophores. What this enables you to do is to look at the localization of different proteins by using fluorophores that emit different signals so that you can see them differentially and therefore look at their spatial profiling. So this is a really beautiful image here by one of our collaborators at the University of Pennsylvania. This is the mouse spinal cord, and you can see clearly here that we've used multiple antibodies tagging different proteins with different fluorophores. So the general immunofluorescence or IHC fluorescence staining protocol starts with a coarse fixation, followed by permeabilization, blocking, antibody incubation, or secondary antibody incubation if you need it, mounting, and then analysis. And alongside these as well, we have to make sure we're doing sufficient washes in between our antibody incubation steps to make sure that we are removing as much of that non-bound antibody as possible to get the best background. But again, this is a kind of whistle-stop tour of the overview of the protocol. I'll break down each step in depth later on. And then this is a bit of a different protocol, but this is for paraffin-embedded tissue, typically used for IHC. So here you can see that we start with our tissue sample before having to deparaffinize and rehydrate. So we remove the paraffin, by rinsing the blocks in xylene, and then we rehydrate with decreasing series of alcohols, followed by enzymatic blocking, and then your typical antibody incubation, and then your signal detection. That's typically using a chromogen such as DAB, before you're then able to visualize your slides with a bright field microscope. So, as I said, that was just a whistle-stop tour, but what are the sort of two typical formats that people use in order to look at the signal? So typically there's fluorescence or there's chromogenic. So why would you want to use one over the other? So fluorescence is excellent if you're looking at multiplexing. So by multiplexing, we mean trying to image multiple proteins in the same tissue. And because of the way that fluorescence works, and you can really easily get non-overlapping fluorophores, fluorescence is really the best for this. You can multiplex chromogenic, but it's a bit harder, and it's also, you have less dynamic range in the chromogenic as well. Fluorescence is also best for target co-localization for the reasons I just said with multiplexing. And it has high dynamic range, meaning that you can see low abundant targets as well as high abundant targets in the same tissue. And if you use a confocal, you can do what's called Z stacking and stitching. So that means you get really high resolution images through the stitching, and then Z stacking means that you can image thicker tissue as well. So that's why fluorescence, if you're using a confocal, is typically better for thick tissue, and also it's really good for cells as well. Cells don't necessarily work that well with chromogenic staining. And then on the chromogenic side, if you're looking at a really low abundant target and that's all you care about, just that one target, then chromogenic is perfect for that. You have a much better signal amplification. You have signal that pretty much lasts forever. So the chromogen, the DAB, for example, you can keep those slides for 20, 30 years and you should still be able to see your physical result. It's also best for FFPE clinical tissue, purely because sometimes this tissue is quite old and can lead to autofluorescence. So that's why we often recommend using chromogenic FFPE in terms of when you have older tissue, because that leads to less autofluorescence, and it's also best for thinner tissue, which is typically again used when you're looking at FFPE tissue blocks. So that was a sort of quick introduction. Next, we're going to go through our protocol step by step and sort of talk about ways that we can tweak things in order to get the best results in your hands. So firstly, starting with protocol preparation, first you want to, your sample preparation, sorry. Firstly, you want to understand your protein function and distribution. You can look at things like your protein expression and understand how that's affected by its cell health and confluency. So in this example on the left here, you can see cells that are mycoplasma positive have much weakened staining, much more disrupted staining in terms of their actin staining. So phalloidin is an actin stain. And then in terms of confluency, say you're trying to look at something like occludin, which is a tight junction marker. You want your cells to be really quite confluent in order to be able to see that marker. So you can see when they're not confluent enough, they're only 50%. You can not really see that tight junction marker that well. But when you're looking at 80 to 90% confluency, when looking at this type of marker, that's really important to see how that the higher confluency leads to much better imaging. And that's sort of what we're trying to say when trying to understand your protein function and distribution. Something, a high confluency for this with occludin doesn't necessarily work for all markers, so do be aware of what is best for your target at the time. And in terms of fixation, when I've been talking to our R&D team about how best to educate our customers and our scientists that watch these webinars, they always say that fixation and sample preparation is by far the most important thing that you can do in order to get the best results. So fixatives and fixation is really important because what it does is it prevents tissues and cells falling apart due to natural sort of degradation processes. So typically the ones that are really commonly used are things like formaldehydes, typically PFA or paraformaldehyde or formalin. And what these formaldehyde fixatives do is they work by acting as a cross-link fixative. So they cross-link proteins and form this insoluble meshwork that means they no longer degrade. And cross-linking is excellent as it conserves cellular morphology and is typically used for tissue. So FFPE stands for formaldehyde fixed paraffin embedded, or you can PFA fix and PFA perfuse animals as well when you're doing tissue collection, and that tends to lead to the best results. One of the main issues with cross-link fixatives like PFA or formalin is that they can mask epitopes and cause autofluorescence. And we'll talk about how to handle this later on, but this is just one of the main issues that people find using this fixative. Other common fixatives that are used are organic solvents, typically -20 ethanol or methanol. And these are really good, especially for cellular, so ICC, cellular immunofluorescence. This is because they don't mark epitopes and they're really good for cells and cell surface markers. However, they can also be damaging to certain epitopes, and this is because you can get a loss of lipid components. So organic solvents essentially work because they dehydrate the cells and then cause the proteins to precipitate and denature in situ. So that's why you get some of these better fixatives, but also some of the issues alongside that as well. So when you are trialing a new antibody and trialing a new sample, do think about playing around with different fixatives, different lengths of the fixation, and also different formats of your fixative. So here's some really nice examples from our scientists. So TOM20 is an intracellular protein that's only located on mitochondria. So you can see here with PFA fixation, really beautiful staining. But when we move to methanol, you're getting a lot more sort of blurry background. This isn't something you want to publish with or present to anyone. So this is just a nice example of switching the fixatives. And then on the flip side, something like E-cadherin, which is an extracellular tight junction marker, you can see that E-cadherin works much better. You get much more clear staining when using methanol than when using PFA. So this is just sort of an example of how changing your fixative can have really drastic effects on your results and lead to really beautiful images that you can see here. The next step in terms of our protocol adjustments is antigen retrieval. So when I was doing a lot of IHC, back when I was doing my PhDs and postdoc, our lab didn't typically use antigen retrieval, but now I know what I know. I think there have been so many cases where I would have got much, much better results if I'd performed antigen retrieval on the tissue. So antigen retrieval is important because what it does is it helps to relieve any protein cross-linking that has masked any of the potential epitopes. And it's typically used in tissue, especially in paraffin, FFPE tissue or PFA fixed tissue. So the most common one is called HIER, so that's heat-induced epitope retrieval. And what this does is it utilizes buffers such as tris-EDTA or citrate buffer. And what these do is they act on, they're sort of like calcium chelators, or they act on the crosslinks and they help to remove the protein crosslinks and therefore reveal any epitopes that were previously hidden. It's quite a gentle epitope retrieval, and it's done typically in a water bath or in a microwave. You would say heat it up for 10 minutes and then let the buffer cool for 10 minutes, and then you're pretty much ready to go. If that isn't necessarily working, then you can try a slightly harsher epitope retrieval. So this one is called proteolytic epitope retrieval. And this is good for more difficult epitope recovery. However, it can damage tissues, so do try the other methods first before you trial the protease one. Typically, you use a protease like protease K or trypsin, and you don't have to heat this up too high. It's done at sort of body temperature, and it has a slightly shorter incubation. So this is just an example of showing the different types of retrievals that you get for heat-induced by looking at different buffers. So when I was doing a lot of tech support, I started Proteintech in tech support. And I remember I had a few customers that would come to me saying that some of their antibodies weren't working. And I said, have you tried antigen retrieval? And they said, yes, I've tried sodium citrate and I'm still not getting any signal. I was like, well, our internal team have tested both sodium citrate and tris-EDTA, and they've shown that tris-EDTA buffer has much better epitope retrieval than with sodium citrate. And so you can show them data like this, and I think it's relatively powerful at showing people how it's really important to not just stick to your one trusted protocol, to actually play around, try different reagents, different buffers, and see what you get. So yeah, I think this is a really powerful message showing that you can try switching up your antigen retrieval and get different results. And it goes the other way as well. Sometimes you get better results with sodium citrate over tris-EDTA. And then this is a different example whereby this protein here responds a lot better to the slightly harsher epitope retrieval technique of the protease K buffer. So you can see here, protease K reveals much better signal to noise, is revealing this epitope much better than with the high background that you're getting when you're just trying sodium citrate heat epitope retrieval. The next point in our protocol optimization and looking at the different steps is permeabilization. So permeabilization is an incredibly important part of a lot of IF and IHC protocols, because what it does is it allows antibodies to access intracellular targets, especially following cross-link fixation. So typically antibodies can't enter the cell membrane. So what we want to do is utilize strong non-ionic detergents such as Triton X. And what that does is it allows you to access intracellular and sort of intra-organelle staining, so say within the nucleus, within a mitochondria, as well as in the cytoplasm. One tip as well is that you can skip the permeabilization step if you have, say, used an organic fixative. So organic solvents like methanol, ethanol, they have already broken down the cell membrane, so you have no need to use a strong non-ionic detergent like Triton X. And also FFPE tissue does not require permeabilization, because the whole process of embedding your tissue into a paraffin block and then it goes through sort of that complex dehydration process, and then the same with the rehydration for staining, that essentially permeabilizes your tissue really well. So you don't need to add Triton X. So this is typically just used with PFA fixed tissue or cells. One important thing to think about permeabilization as well is that if you're looking at, say, thicker sections or organoids or spheroids, you really should think about increasing your permeabilization, so slightly increasing the concentration of your Triton X and increasing the time. This is something you should play around with, because sometimes leaving your tissue too long in the Triton X can cause it to degrade a little bit. I've definitely made that mistake myself in the lab. And then also one thing that I've learned since joining Proteintech, and sort of I've been here 3-4 years now, I've learned so many tips. And one of them is that antibodies can actually get stuck intracellularly, and that then will cause high background. So one way that you can sort of, if you think this might be the case, if you've got a relatively thick sample, or you think that your antibody is getting stuck, then one thing that you can do is to increase your detergent concentration and its duration, and also increase the number of your wash steps. So it's all very well doing slightly longer wash steps, but increasing the number of your wash steps is what's really important at trying to remove any unbound antibody, and any of that unbound antibody that might be getting slightly stuck intracellularly and causing high background later down the line in your workflow. The next step for our protocol optimization is looking at blocking. So protein blocking is one of the typically things that we do for immunofluorescence. And this is really important because what it does is we use things like serum, so either donkey serum or the serum that your secondary antibody is raised against, or BSA, which is bovine serum albumin. And serum is really important. It's a good blocking reagent because it contains antibodies that bind to non-specific sites. If you are using BSA, then it can be a good idea to also include Triton, because what this does is this acts as a surfactant, and it helps to aid in the wetting of your sample and improve the coating of the BSA. The next part of blocking is enzymatic blocking, especially if you're looking at IHC and using a chromogen, then this is where enzymatic blocking is really important. So what this does is this presents any high background or false positive signal. So say you have a secondary antibody with HRP as its detection reagent, you want to make sure that you're blocking any endogenous enzymes that could be activating this HRP by using H2O2, which is hydrogen peroxide. If you're using AP, which is a slightly less common enzyme, alkali phosphorate, oh, sorry, I can't say that word today. You can block that with levomisol or with 1% acetic acid buffer, and that should prevent any endogenous enzymes causing higher background. One tip that came about from our R&D scientists when they were developing some of our IHC kits is that they discovered that there's really no need in the majority of cases with FFPE sections to do peroxidase quenching. So we have slides and slides, so much data demonstrating that there is no difference in the background with various antibodies when you're looking at quenching versus not quenching. But I will stress that this enabling you to skip the step is obviously great. If you have FFPE sections, it will save you time. It will also save you a perhaps slightly damaging process as well. But if you are looking at frozen tissue or anything else that with a chromogen that isn't just FFPE, then it's really important that you have this quenching step. So frozen tissue obviously hasn't gone through the whole paraffin embedding and dehydration process, but clearly has deactivated any endogenous enzymes. So again, this is just a fun tip if you are using FFPE sections. The next thing we want to think about when we're performing our steps is what kind of primary antibody staining we're going to do. So typically, in the one that most people are aware of is the indirect staining. So this is where you have one primary antibody that's unlabeled and that binds to your epitope of interest. And then you detect that primary antibody with a secondary antibody that has your detection conjugate. So that might be either a fluorophore or a chromogen, like HRP. And this is, again, sort of something that most people use, but more and more popular now is the direct method. So direct is where you have your primary antibody pre-labeled with a fluorophore. And what that does is that then skips any of your secondary antibody steps, so it's saving you time, but also it's enabling you to have slightly better resolution because you're not having any off-target binding. One thing to note, though, is that if you do have a low abundant epitope, indirect staining is definitely much better because what can happen is you can get 4, 5, 6 secondary antibodies binding to that one primary, and that therefore has much higher signal amplification and will have better sensitivity for yourself. But why would you want to go direct? So as I said before, this is how you do it using a pre-conjugated antibody with a fluorophore or chromogen or self-label. And it has quite a lot lower background, so you get slightly higher resolution images. This is because you don't have any off-target binding from your secondary antibody. And this off-target binding from a secondary antibody can be really problematic for some customers. And it used to be in myself. I did a neuroscience PhD on a lot of mouse tissue. And if I was using mouse monoclonals, sometimes I would get really high background from your mouse secondary antibody binding to endogenous receptors in your mouse tissue. However, that is not the case if you go direct because you are eliminating the need for secondary antibodies. So again, this is something to consider going to direct when you have mouse-on-mouse staining problems. Because you're omitting the secondary antibody, the protocol is much faster and simpler, and you can have really easy multiplexing because it enables you to use antibodies from the same species, again, because we're able to omit the secondary antibody. I've had a lot of customers after presenting these data with our primary conjugated and antibody labeling kits asking me how can they multiplex labeled and unlabeled primary antibodies. So that's sort of suggesting in this diagram, can I use a primary, unlabeled primary and secondary alongside my label conjugation? And the answer is yes, definitely. It works really, really well. There are just some things you have to be cognizant of. So if your primary antibodies are all different species, then that's fine. You can label all your primary antibodies together and then incubate a secondary as normal and image. However, if your primary antibodies, the labeled and the unlabeled, are of the same species, then first what you want to do is run your unlabeled and labeled secondary as normal and wash very, very well. So you want to remove any of that secondary antibody that might be binding to your other primary. So you want to remove that, wash really well, and then you can simply incubate with your labeled primary and then you're ready to go. So it's obviously a slightly longer protocol, but it still gives you the results that you need, the really beautiful labeling. So that's it for the protocol considerations. Next, we're going to look at the reagents available to you for immunofluorescence and for IHC. So as we're an antibody company, and because immunofluorescence and IHC is an immunoassay, choosing the right antibody is an incredibly important part of getting your best results. So one thing that I'm not sure people are always aware of is that not every antibody is suitable for every antibody application. So do make sure you check your supplier's validation data. For example, this is one of our antibodies that is only suitable for Western blot, and this is probably because it only recognizes a linear epitope. So as we know, when we run a Western blot, we denature the proteins by SDS page, so the protein becomes linear, and it might be that these Western blot only antibodies are only recognizing linear epitopes. Therefore, they are not suitable for IHC. whereas some antibodies are suitable for sort of all applications because they can recognize proteins in all of their conformational states. So that might be its native conformation, like you need for IHC and flow cytometry, and it also might be its Western blot. So that's just something to think about. And in terms of your primary antibody type, there are three main types of primary antibodies. So the first one, and the one that the majority of antibody users use, are the polyclonal antibodies. So the polyclonal antibodies are a mix of IgG. And what this means is that they are really better for low abundant targets because they have increased sensitivity. This is because they're more likely to react with different epitopes across your target protein. They're better for cross-linked fixatives. In case that cross-link has hidden the one epitope that's available, you've then got other epitopes that are able to be detected by a polyclonal. Better for cross-reacting with rare species that might have lower homology with the, for example, the human protein that was the immunogen for the antibody. And they're better for mutated proteins. Say you're looking at, I don't know, say in neurodegeneration, you've got a protein that's been truncated. You're more likely to react with that if you've got a polyclonal antibody recognizing multiple epitopes. Some of the disadvantages are that you do have high lot-to-lot variability and also high background due to the fact that, again, you might be recognizing off-target proteins because of that homology with the multiple epitopes. And then with monoclonal or recombinant, as the name suggests, you're only recognizing one epitope. So if you're interested in epitope is 5 to 8 amino acids long. And these antibodies can only recognize one because of how they're made. So these are much better for high abundant targets, but they're also really good if you're struggling with high background with a polyclonal, then we really recommend switching to a monoclonal and a recombinant. Other things as well to think about is that they are much better for reproducibility, especially recombinant antibodies. They're excellent for long-term projects. There's going to be no change in the lot-to-lot consistency, and similar with monoclonal, there's almost no change lot-to-lot. So if you have a long-term project, then I definitely recommend trialing a monoclonal over a polyclonal to see if that works for you. And in terms of what Proteintech can offer for you, especially with the direct kind of staining, we offer a lot of our really popular antibodies are now available directly conjugated to our Coralite dyes. So Coralite is essentially our version of the popular Lexaflor. They are equivalent in performance, so they're available in these 4 popular colors. They're also compatible with cyclic immunofluorescence protocols. And you can see here some of our collaborators. So Derek Sung at the University of Pennsylvania and Łukasz Krobok at the University of Bristol. They've produced some really, really beautiful images of these. So something I definitely recommend trying because you get these really excellent, easy, fast protocol with excellent high resolution and lower background. But one thing as well that we're really excited to present to you as a company, and that has been, I think it's been released for over a year now, so you guys might have heard of it, is our flexible antibody labeling kits. So what these are excellent for is that they're really flexible, as the name suggests, because they enable you to multiplex antibodies from the same species, but they're also compatible with any primary antibody. It doesn't have to be an antibody from Proteintech, and it only takes 10 minutes to label. So previously, if you wanted to self-label or pre-conjugate an antibody yourself, it could take many hours in the lab. You'd have to use something as high as 100 micrograms of antibody, and you'd have to do buffer exchange so that it was PBS only. Our flexible antibody labeling kit works in any buffer, and you can label as little as 0.5 micrograms in just 10 minutes. We have them now against all these different rabbit and mouse specificities, as well as these 7 different fluorophores, and now also these enzyme conjugates, so biotin and HRP. This image here is really beautiful. This is using 3 mouse monoclonals, so you can see how easy it is to multiplex. As I said, the workflow is only 2 steps and takes just 10 minutes. So first you have your primary antibody, which you incubate with the flex linker and some flex buffer. Then you just simply add the flex venture to remove any of the unbound linker, and then your antibodies are ready to use and you can get really beautiful staining. If you want to multiplex antibodies from the same species, then that's really easy. You just have your 3 different antibodies, or 4, as many as you want, fluorophore permitting, of course. And then you label them individually with your kits, as I outlined earlier, and then mix them together and add them to your slide, and you're ready to go. So you can see here, got multiplexing of 2 mouse monoclonals in this immunofluorescence image. And you can see it was really quick and also really beautiful imaging. And the beautiful thing about multi-flexible is that, again, as the name suggests, you're not limited to just using it with one antibody. You can use it with as many antibodies as that specificity allows. Therefore, you can have really flexible panels for immunofluorescence. And then while we're still talking about kind of immunofluorescence and the reagents for it, the next really important point to think about is the fluorophores. So there are typically two types of fluorophores when looking at this. So we've got our synthetic dyes. So our synthetic dyes include things like our Coralite Plus dyes, which, like I said, are the equivalent of the traditional Alexa fluor. And what these are sort of traditional organic dyes that have been sulfonated, which means they're negatively charged and more hydrophilic. They are also brighter, pH insensitive, and last a lot longer. So that's why these synthetic dyes have sort of become really huge in the life science reagent market because they are so reliable, they're so bright, and they're not really affected by many experimental conditions. That's why they're really, really popular. And what I'm talking about is over the sort of more traditional dyes like FITC and Texas Red. These dyes nowadays as well, their chemical formulas, they've become a lot, dye makers have become a lot more clever at making sure that these are really sustainable in terms of signal, but they still are not as good as the synthetic dyes. I remember when I was, during my PhD, I was using FITC to do some demonstrating for an undergraduate practical, and we were using FITC for looking at the brainstem, I believe. And I could only show 2 or 3 students this one image before it would just completely dim, because that was as far as it would allow me to use. So that's just something to bear in mind. They might be slightly more expensive than your traditional dyes, but they are definitely worth it. One tip as well that I got from our scientists at Proteintech is that using the correct mounting media is really important, because what that does is it helps to optimize the refractive index and also slows down any photo bleaching. So there are lots of different types of mounting media out there, so do have a look and see which one works best. Some mounting media works better for certain brand of fluorophores than others, so that's just something to be cognizant of, and it's definitely a cheap switch to help improve your imaging for sure. Whilst we're on the topic of sort of fluorophores, we're going to then talk about secondary antibodies. So secondary antibodies are, as you all know, typically the thing that helps us to detect our unlabeled antibodies. So first off, you want to ensure that you've got the correct species for your primary. One thing people might not be aware of is that you have different mouse monoclonal isotypes. And what you can get as well is isotype-specific primary and secondary antibodies, or you can get generic mouse secondary antibodies. So you want to look for something that says the heavy and light chain, or you've got the isotype specific in terms of how you're detecting your mouse monoclonal. The other thing you want to check with your secondary antibodies is that you have non-overlapping fluorophores. This is really, really important because you might not be aware that things like FITC and Coralite fluorotate, they actually have almost identical spectra. So you can use things like SpectraViewer. We have SpectraViewer on our website, or there's SpectraViewer available. It's a free tool that enables you to plug in the fluorophores that you're using, and it will show you the spectral overlaps. You'll know whether or not your fluorophores are suitable. If you are multiplexing with secondary antibodies, then we do recommend you choose highly cross-absorbed, because what this means is that you're then unlikely to get any cross-reactivity of your secondary antibody cross-reacting with, say, an antibody against the species that it's not designed for. And then in terms of IHC secondaries, one thing that we really recommend and always use ourselves at Proteintech for the chromogen side is the polymer HRP. So polymer HRP is really clever. What it is it's a specialist depth strand backbone with the HRP attached to that. And then a lot of different biotin-related secondary antibodies are able to attach to that. So what this means is you get really excellent signal amplification. And then compared to the sort of streptavidin type, traditional streptavidin dyes that you also used to get for HRP, these can, the problem with those is that they used to really badly aggregate and cause high background. Whereas the nice thing about polymer HRP is that you get less aggregation, but you still get that really excellent signal amplification. So like I said, it's something that we always use at Proteintech, and it's something in our tech support team we always recommend to customers as well. We've also literally, I think last month, just launched our multi-Rab recombinant secondary antibodies. And so what these are is a multiclonal mixture of recombinantly engineered antibodies. So as I touched on before, recombinant antibodies are monoclonal in nature, but what we've done is we've chosen monoclonals which react to different parts of your primary antibody, so either your mouse or your rabbit, and that means that you've got sort of really high lot-to-lot specificity. We've got kind of engineered specificity as well because of the recombinant nature of them. They are definitely outperforming some of the competitor secondary antibodies that are not recombinant, and they also have really low cross-reactivity with off-target species. So this is something we're really excited to launch. Yeah, this is definitely something to consider. We will include links to the multi-Rab page after this talk so that you guys can find out more for yourself afterwards. Oh, and I am sorry, this image isn't coming up, I'm not sure why, but the nanobodies as well from our sister company, Proteintech, these are really excellent for things like tissue clearing. So nanoboosters, what they are is they're a small nanobody that has a fluorophore attached. And because it's 10 times smaller than your traditional antibody, what this does is enables huge reduction in incubation times due to increased tissue penetration. And also you have much lower epitope label displacement because of the minimal linkage error. So customers that do tissue clearing, so things like VDisco, a lot of neuroscientists do it to visualize some neuronal tracking. And that's what nanobodies are really excellent for. And if my PowerPoint was working, that's what you'd be able to see in this image. And I've just sort of included a kind of cheat sheet here, lastly, to talk about the different types of antibodies that are available to you and which is best for you. So with our flexible labeling kits, they're really good for same species multiplexing, same for our correlate pre-conjugated antibodies. But in terms of the traditional multi-Rab secondaries, you have to use different species multiplexing here, but that's not to discount them at all. If you're looking at mouse-on-mouse staining, then directly flexible or correlate pre-conjugated are really excellent for that. Secondary antibodies can have issues, can have not. That's why I've put this, the question mark. And in terms of how abundant is your target, which one should you use? If you have a high to low or high dynamic range, then using traditional is really perfect for that. Correlate pre-conjugated are also really good for high to medium low abundant, and the flexible antibody labeling kits are good for high to medium abundant targets. So just think about when you're choosing what signal development method to go for, think about how abundant your protein is in your cell or your tissue before deciding which one to go for. And in terms of panel flexibility as well, obviously with secondary antibodies and with flexible, you've got much better flexibility because you're not stuck with one fluorophore attached to that primary. Whereas with correlate, you have only got one fluorophore, but that, if you know you're always going to use it in that panel, then correlate is perfect for that. So the last part of my talk is just going to go through some detailed troubleshooting. So first off, when we're looking at immunoassays, we really want to think about the controls. So controls are so important because otherwise you're just looking at signal, and you have to make sure that what you're watching is, what you're seeing is real biological signal. So ways that we can do that is we can test our secondary only. So what we'll do is we'll emit the primary antibody from the buffer, but run the workflow as normal. And what this does is this will ensure that the signal detection, or the signal that we're viewing, is specific for the primary antibody binding and not just the secondary antibody binding. The next thing we can do is our endogenous control. And what this is when we will remove all primary and secondary antibodies. And this is especially important in immunofluorescence because it reveals any level of autofluorescence that might be occurring or any artifacts that might be looking like real biological signal. The next step are our antigen controls. So our antigen controls enable positive control. So that would be using cell or tissue that is known either by yourselves or in the literature to express the target protein. This confirms that your protocol is working and also will validate any negative results that you might be seeing. The next one is the negative controls. That might be like a CRISPR-Ko cell line or a mouse knockout of your target protein. By running your antibody with this, you'll show that the antibody is specific for the target. If you're short for time and you have a new antibody or a new tissue or cell type that you're trying out, then I really recommend you at least run your secondary only and your endogenous control, just to be sure and really cognizant that your antibody and your results are working well. So one thing I've had when I've done this talk many times, is I've had some customers coming up to me absolutely devastated because they have really high autofluorescence. When we were in SFN last year, we had a customer who really didn't know what to do. The autofluorescence was really high. So these are some tips that I can sort of give you to try and help with your autofluorescence. Sometimes there really isn't much you can do. But one thing to start with, and as I mentioned before, is to always include an endogenous staining control. This will sort of really reveal the background autofluorescence of your sample and will help you to determine exactly what you're working with and how much you need it to decrease by. One thing as well that's really interesting is that the majority of autofluorescence from various causes, such as paraformaldehyde fixation or heme in red blood cells, this all occurs in the green spectrum. So one thing you could try doing is switching to far red and red fluorophores to sort of try and minimize that as much of the autofluorescence as you can. So we've got our Coralite Plus and our Coralite 594 and 650. So these are really excellent fluorophores that you could try. We also have these in our flexible kits if you're interested in trying to minimize autofluorescence. And as I said before, it's fixation, so cross-links fixatives and red blood cell heme that causes a lot of the autofluorescence issues. So that's why fixing your samples to the minimum time and also perfusing your samples with PBS if you can will really help to reduce autofluorescence. One issue that can cause autofluorescence is age. So say you're looking at clinical samples that are really quite old, so old patient samples, or they've been in the paraformaldehyde or the formalin for a really long time, something that you have no control over. That can be a massive issue and cause high autofluorescence. So you can then try and use sort of general blocking reagents like Sudan Black B. And there's also other, from competitors of ours, they sell really excellent autofluorescence quenching kits. So do check them out. We know they work really well. So just something to think about if you are really struggling, like that customer was from the conference. So as well as autofluorescence, we can think about how to troubleshoot weak or no signal. So typically the causes of this might be something like the antibody has lost activity. So you could try a new lot or check its storage as well, make sure it's still working. It might be that you haven't fully adjusted your antibody concentration or your conditions. So try titrating your antibody concentrations, adjust incubation times, try things like increasing the permeabilization, especially if it's an intracellular target. If you think your protein of interest is not expressed, then do run a positive control. Like we said before, this confirms that the antibody in your protocol is working in your hands. If your protein has low expression, try single amplification, and also try epitope retrieval if you're using a cross-link fixative. Next step to try and troubleshoot is high background. So again, high background might be due to an incorrect antibody concentration use. So again, try doing a titration step and go through as many titrations as you need in order to find that excellent antibody concentration that allows you the best signal to noise ratio. If you think it's an issue of non-specific antibody binding, then do prolong your blocking step and also increase the concentration of your blocking solution. Poor sample washing is also a massive cause of high background. So I definitely recommend increasing the number of wash steps. Maybe say you're doing 3 5 minutes, you can increase to 5 minutes. What that does is you're just increasing the mechanical force of the PBS, removing any unbound antibody and potentially causing any high signal. And then the other one is if you think your slides are drying out, then try using a hydrophobic pat pen or a humid box to prevent that happening. Talk to people. There are lots of kind of really clever methods online. If you can't afford a proper humid box, there are lots of ways that you can make it yourself quite cheaply. I definitely recommend trying to improve on that, because slides drying out is honestly a very big cause of high background. And this is one problem that I used to face as well. I remember once watching all my brain slicers float down the drain. So I know that this can be a big issue for some customers. Sections dissociating or coming loose. This can be caused if you're not using charged slides, or if the slides you're using are out of date. I wasn't aware that slides could go out of date, but make sure that you're using relatively new boxes. Or if you can't afford to charge slides, then you can also make your own by coating them in gelatin. There are lots of excellent recipes out there online, or let us know and we can send you one of ours. If your slides are not properly dried when frozen, that can also cause massive issues with staining later down the line. So do make sure that you are defrosting them and drying them fully for 30 minutes before then fixing and staining. Sometimes distilled water loosens your slides, so try using a PBS wash buffer, and also antigen retrieval. Sometimes that can cause slide dissociation, so try different methods. So say you're using a water bath, try a microwave or vice versa. And then lastly, if you are doing IHC and you're having a problem, we have spent a lot of time and effort as a company developing our own IHC kits. So we have our IHC Easy kits, which contain everything you need, including the primary antibody. And these have been properly optimized in order to get perfect results first time. Or if you would like to have the flexibility of swapping out which primary antibody you have, then our Prep and Detect IHC kits contain everything you need, including our recommended antigen retrieval and all our wash and blocking buffers as well. If you're interested in what you've seen, then you can definitely check out our other videos online. We've got image analysis workshops, we've got controls for IHC and IF, as well as other antibody applications. And if you're not aware, this talk as well is part of our larger immunoassay week. So tomorrow, my colleague Frida is giving her talk on immunoprecipitation. And then on Thursday, we also have a talk by our lovely Amy, who is giving a talk on Western blot. I'll keep this up for a little bit, but if you are interested, you can apply for a free sample. You can scan the QR code here with your phone, or we will send everyone out a link at the end of this webinar so that people can apply for their free sample themselves as well. And I think with that, I think Frida will come online, and I can see that there have been lots of questions. So
yeah, there's some great questions. I've been reading through some of them. There's one that I think that would be a good one to start with. So Maria is asking, how would you adjust an IF protocol to visualize surface proteins and intercellular or transmembrane proteins?
Yeah, that's a really good question. And I think, unfortunately, there isn't a simple answer for that, but I think it depends on the intracellular protein and the extracellular protein that you're looking at. But I would definitely first start with the fixative, see which one works for you could try them individually. So you first want to look at your extracellular and your intracellular protein sequentially, and then see perhaps like PFA looks good for the intracellular, but bad for the extracellular, but methanol also looks good for the intracellular and for the extracellular. So then you therefore know that, okay, methanol is the way to go here, and kind of go down that way, try and figure out what types of protocols are best individually, and then hopefully they will merge together.
Yeah, and kind of to add to that too is, sometimes you also kind of want to look at your buffer too, because in certain situations, detergent can be your friend, and in other situations, it can be really detrimental. So if you're looking at more of an extracellular protein, maybe you start off with less detergent and then kind of titrate and work from there and see, what detergent concentration is good for your buffer in order to see your protein of interest. But yeah, with a lot of these questions, it's like you wish that there was the perfect answer, but a lot of times it's not. It's a lot of trial and error.
Definitely.
So we have another question that says, how do you prevent tissues from getting detached from slides when you're boiling interest?
That's a good question. I've never had that issue myself, but there are sort of maybe different ways that you could try. If you could try perhaps you choosing a special slide. So if you're not using charged slides or gelatin coating slides, then try that. You could try either using a microwave or a hot plate and switching between the two. I don't know, Frida, if you have any other suggestions.
No, those would be my suggestions too. I did have a lot of issues with my experience with samples coming off my slides, which is like so disheartening because it takes so long to, get everything prepared and do all the sectioning and stuff like that. For me, the best was dipping my own slides in gelatin. So I know there's a lot of commercial slides available, but there's also a lot of great protocols out there on how to like dip your slides in gelatin. And so that's what worked for me
Yeah,
personally.
I found gelatin better than the charge slides as well sometimes.
Yeah,
Yeah.
yeah.
It's all about trial and error, unfortunately. Because one thing, something that works for someone might not necessarily work for someone else. And the same for the different proteins that you're using. One fixation might not work for the other. So it's just about kind of trying to get that play off to find out what is sort of least detrimental to the other.
Yeah. So there's another question about antigen retrieval. So which antigen retrieval method should you try first if, you're seeing that your antibody stain isn't ideal?
That's a really good question. So we find typically that Tris EDTA buffer at pH 9 works in the majority of cases. Sometimes sodium citrate buffer at pH 6 is better, but the one that we run hundreds of thousands of IHC over the years, and the time I'd say 8 times out of 10 we recommend Tris EDTA. And then if neither of those are working, then you can try the proteolytic retrieval method. And then if you're still not seeing anything, you then might want to consider, if you can, a different fixative. Again, that can be really hard, especially if you're in tissue. But I know some clinical samples, they are moving now towards using, I think, acetone as a fixative. So there are lots of different fixatives out there. Also look at the literature, see what your colleagues are doing in sort of similar experiments and similar tissue. How are they preparing their samples? As I said, like everything that I've sort of learned over the past few years at Proteintech is that sample preparation is by far the most important thing that you can do. The kind of workflow down the line from there, you can do little tweaks, but you have to ensure that your sample is sort of prepared in the best way. So that's either isolating it really quickly and fixing it in sort of the correct fixative, a sufficient amount of fixative as well. And then the staining down there is obviously very important, but it's not as important as the kind of quality of your sample and the fixation steps of your sample.
Yeah, that's really great advice. And especially, the looking through publications, it's like there's no need to, reinvent the wheel again. And so, yeah, it is really important to kind of see what others have done and what's worked for them.
Yeah, and a customer just said actually that they had issues with tissue boiling of slides before, and that they found the microwave was too aggressive, so the water bath actually worked better for them. So thank you, Leonie. That's sort of a nice example of switching things around and actually finding that what works best in your hands.
Yeah, and like sometimes it's the smallest things, like the things that you wouldn't think would actually change anything. Like it's not necessarily, changing reagents, but the apparatus you use to heat, or it's very interesting how like these things happen in the lab.
Yeah.
So we had a few questions asking about kind of how to minimize fading. So if we have any recommendations on kind of mounting solution.
Yeah, so fading, again, the type of fluorophore that you use is really important. So we've recently switched from our Coralite to our Coralite Plus fluorophores. And the way that these are is that they, correct me if I'm wrong, Frida, I think they're a bit more pegylated, or they've got a slightly different chemistry that means they are brighter and also less sensitive to fading. And the other point to note is that there's many different types of brands of mounting media out there. And we've had feedback from our customers before that Coralite works really well with certain mounting media and not with others. And they said it was really funny that the Elixifluor fluorophores work much better with one, but with rubbish with the other. So I definitely recommend, if I remember correctly, mounting media isn't that expensive. So do try maybe try a range of different brands of mounting media, see what their ingredients are if they disclose that, and play around with that and see if that can really help with fading. But yeah, definitely the point of fading is the type of fluorophore that you're using and also the mounting media.
Yeah, me personally, I've had really good luck with like hardening mounting media. I used to use like the, anti-fade mounting media all through graduate school. And then once I went to my postdoc, I started using like the hardening mounting media, and it was amazing because it was just like weeks later, I would still see really good staining on my tissue. So,
That's
like
really,
Rebecca said, I think it depends on not only like the tissue, but obviously the targets I was looking at were different too. So it probably is a culmination of all these
yeah.
things.
I spoke to a microscope vendor once, and they said that it was also like the how you dry your tissue, I think he said was really important. Um, but again, it's I don't know, he was looking at a certain type of tissue that we weren't working on. So we were working on brain, and we tried loads of stuff to try and prevent fading, and it wasn't working. But I think he had like a colon section. So I think again, it just depends on the makeup of your tissue, so many different things, the phase of the moon, joking, but
Yeah, right. Sometimes it feels that
it
way
feels
though.
like that sometimes, yeah.
Um, So we also had a lot of questions about autofluorescence. I know you touched on it a little bit in terms of some tips, but there was a few people who were talking about working in heart tissue, and of course this would be an issue when it comes to autofluorescence. So do you have any other tips or recommendations on how to kind of manage autofluorescence in these types of tissues?
Yeah, I guess I can only kind of reiterate what we touched on before. So autofluorescence is caused by three things predominantly. So it's caused by cross-linking fixatives. It's caused by red blood cell. The molecule heme is really strongly autofluorescent, and it's caused by something called lipofuscin, which is a molecule that accumulates, I think, in vesicles over age. So if you have, unfortunately, like human clinical heart tissue that's really old and has been sat around in formalin for ages, then you are unfortunately going to have quite a lot of high autofluorescence. Um, so things that you can try and do if you're, say, working in animal tissue is that you can perfuse your tissues with PBS before then perfusing with the PFA. I used to do that with mouse brain, and it was always providing the best results. You really want to be careful as well of how long you leave in PFA. So the amount of time that you leave in PFA and the amount of fixative that you have is determined sort of by the tissue that you're looking at, and also whether or not you perfused it beforehand. So some people aren't aware actually that you actually need a really high volume of PFA in order to make sure that it's really well fixed. But if you have enough, I think it's something like 50 to 100 times volume to the size of sample of PFA that you're meant to use. I think that's something that our R&D scientist Abel said. And another tip is, so ensuring your fixative is done well and done for the least time needed. Perfusing, and then there are lots of really good commercial reagents out there. So there's one called Vector TrueView. That's something I used in the past. It has I think Sudan black B and borohydrate as the reagents, and a mix of other things. And you can look them up. Myself and other collaborators have used it, and it's worked really well. So I don't think it's prohibitively expensive, but you can also make your own if you can't afford that. There are protocols on sort of like ResearchGate that use Sudan black B and other kind of blocking reagents that help.
Yeah, I absolutely agree with everything you said, and especially if you have the ability to, perfuse the tissue beforehand. There's a lot of tells during the perfusion. So obviously, a bad perfusion is going to lead to a lot of autofluorescence. So when you're perfusing, you want to make sure that liver is white, white, white before you kind of even add the PFA. So when you're first kind of fleshing everything out with PBS, there's a lot of tells to make sure that, your perfusion is working properly. And
I guess for
so
cardiac tissue, you've kind of got in from there. So I don't know if you perfuse it. Frida and I are both neuroscientists, if
yeah,
you couldn't tell. we're not
we perfused
necessarily.
everything was perfused.
Yeah.
So I think we have time for one more question. So I thought this was a really nice one. So do certain proteins have a higher risk of epitope masking during fixation?
That's a really good question. Probably, but I'm not aware of which ones, unfortunately. Maybe the slightly bigger ones. Just thinking about it. I don't know, Frida, are you?
So yeah, so it definitely depends on the protein. Larger proteins, like Rebecca said, do have a more risk of, epitope masking just because they are so big. So I guess more can go wrong. So they can kind of, change shape, and also phosphorylated proteins or other proteins that have kind of post-translational modifications, they tend to be more susceptible to in terms of epitope masking. But yes, it's all kind of dependent on the localization of your protein and what your protein is. So yeah, yes
Yeah.
is the
But yeah, so also don't forget about antigen retrieval. Like I said, we didn't really use it enough in our lab, and from what I've seen since joining Proteintech, it's literally night and day. Like the signal to noise ratio is increased so much that your results look amazing.
Yeah. Well, I think we're at the top of the hour. We had so many great questions, like we always do, from everyone. But yeah, I don't know if Rebecca, you wanted to say the final words?
No, thank you so much for tuning in, everyone. And I saw that some people said that you haven't received the email from yesterday. Sorry about that. We had some people off yesterday and this morning. So you should have received the email soon, if not by the end of the day. And the same for this one. There's some processes we have to go through before we can send the email out. So it does take 1 or 2 business days. But you will all get an access to the recording. And I'll also include the recording of the antibody validation webinar yesterday. And if you are interested on joining our other webinars, so we've got the immunoprecipitation one led by the wonderful Afrida, who's on the call right now. And then that's on Wednesday. And then we've also got our Western blot one on Thursday. So yeah, really exciting week. Looking forward to seeing more of you again. So yeah, thanks for joining. Bye.